deep within the oceans and the rich ground below, to the endless fields of crops, and even on our favorite friends, microbes are everywhere. They're naturally found in our bodies. We can find them on our skin, in our nose, and in our guts, where there are at least as many as the cells in our entire body. And about gut microbes, I'll be talking to you today. But how do we benefit from them? Microbes in our gut help us with profitable food fermentation, such as that of the fibers found in vegetables and fruits. They also produce compounds that we cannot synthesize ourselves, like vitamin K. And they help us stay healthy by leaving no space for pathogens or by showing them off to our immune system. Each one of us has a unique gut microbiome, as unique as our fingerprints. And the process of getting our unique gut microbiome starts from the moment we are born. By entering the world, we start taking along microbes from our mothers and the environment around us. The composition of the infant gut microbiome relies a lot on these environmental factors, but mainly on feeding. Breastfeeding is a suggested form of infant nutrition by the World Health Organization. That is because it's beneficial for infant health, with lower occurrences of diseases like asthma and eczema, a lower tendency for obesity, and better cognition development. However, as of December 2023, the World Health Organization estimates that only 44% of infants are exclusively breastfed up to the age of 6 months. This is a highly pressing issue because first-generation infant formula has been associated with allergies, eczema, and obesity. This means that by harnessing optimal infant nutrition, we can safeguard and improve infant health. And this also involves the infant gut microbiota. This means the microorganisms that live in the infant gut. That is because human milk, the golden standard of infant nutrition, not only feeds the babies, but also the bacteria in the infant gut. And let me show you how that is. Human milk contains certain sugars that are called human milk oligosaccharides. And here on, I will show them to you as HMOs. These sugars reach the infant gut almost undigested, and there they can be utilized by certain bacteria that can degrade them. Some bacteria can degrade the sugars, and others cannot. We hypothesize that the bacteria that cannot degrade the sugars collaborate with the bacteria that can in order to survive. However, we don't exactly know how this teamwork leads to the formation of the infant gut microbiota. And to understand this better, we build synthetic communities. These are selections of bacteria that are naturally found in the gut of vaginally born breastfed infants that we put together as we try to model the infant gut microbiota. We like to think of them as construction materials. Just as in a real construction site, we put together different materials to make a bigger, complex structure. And of course, building something from scratch is not a direct process. It needs investigating how each one of the materials fits with the others to make intermediate structures before finalizing the complete complex structure. In the same logic, we start by looking into how different bacteria found naturally in the gut of vaginally born breastfed infants degrade these human milk oligosaccharides. We put together bacteria of different capacities, and based on their interactions, we create our synthetic communities, which we then use to test how human milk oligosaccharides beneficially impact the gut microbiota. Up till now, our research has shown that synthetic communities are an accurate model for the representation of the infant gut microbiota. Not only there is community survival in human milk oligosaccharides, but also following our hypothesis, there is collaboration between the bacteria that cannot degrade human milk oligosaccharides and the bacteria that can, in order for the first to survive. Also, our synthetic communities are able to match the infant gut microbiota in terms of composition, compounds produced, and bacterial functionality. This means that the future holds many exciting new applications for synthetic communities. They could be used as research models to test new food supplements or food formulations. They could even be used to model microbiomes from other ecosystems, such as those from Alejandro's project, from soil and plants. And they could even be used as food supplements themselves, which will make them probiotics. And with the addition of a compound that stimulates them, they could be administered as symbiotics. And this would be to promote infant health, for example, by supporting food digestion or to even cure disease. Thank you.